Hello, everyone. My name is Dina Fanan, and I'm the Director of Product Management here at Biolog. And I'm going to let Max quickly introduce himself as well.
Hi, thank you. I'm Max Gravener. I'm one of the field application scientists here at Biolog.
So, Max and I are really excited to do a different type of webinar today where we share some tips and tricks for phenotypic microbial identification. So, just a couple of quick housekeeping items first. We are recording this webinar, and the link to the recording will be shared with everybody who registered. And then you can submit questions via the Q&A chat feature. And many of you have already submitted questions when you registered. And so we will do our best to start with those and, either answer them during the presentation or whatever we don't have time to get to, we will reach out to you after the webinar. So, fun fact, I recently learned that instead of using fingerprints to identify a person at a crime scene, in the last few years, forensic scientists have been exploring the possibility of using something called the touch microbiome, or basically the unique community of microbes that live on a person's skin. So that's a really interesting idea, and in some ways, it's actually the opposite of what we do at Biolog. So we identify microorganisms based on their unique fingerprint or pattern that they create in our special test plates in order to identify that individual species of organism. And so at Biolog, we actually celebrated our 40th birthday this year, which is really amazing if you think about how long this technology has been around and how it continues to bring value to, performing identifications and just contributing to our understanding of microbes. So what our age also means is that we've gotten pretty good at helping troubleshoot because we've seen a lot of different pitfalls and common mistakes. And so, we've gotten better at helping our customers over the years. And so here's a brief outline of the things that we're going to cover today. So I'll start by giving an overview of phenotypic microbial ID for those of you who aren't as familiar with how it works. And then I'll talk a little bit about our database and how it's used. And I'll go over some hardware options for different throughput needs and also how our software works with the systems. And then Max, who's one of our experts at helping users troubleshoot their work, and he's also collected feedback from our global team of FAS that support our customers all around the world. He will come in to discuss the ins and outs of our various ID plates and our protocols. And then we will open it up for questions, both the ones that you've already submitted, and also, as I mentioned earlier, you can feel free to submit via the Q&A box. And as always, if we run out of time, we will follow up with you after the event. So you probably know that there are many different ways to identify a microbe. So besides phenotyping, which in itself encompasses several different methods of, looking at the color, the morphology, a lot of different ways of phenotyping, there's also sequencing methods and proteomic methods using MALDI. And the good news is our lab services group can actually perform any of these methods for you. So if you only intend to run, a few samples a month, for example, just reach out to us for more information on how you can outsource the work to our lab. But if you want to do IDs in-house, there are some considerations about which method or even which combinations of methods are the best for a given situation. And these could include things like the cost of the equipment, how much technical expertise you need to run it, and also analyze the data and get your answer, how long it takes to get the final result, and then the relevance and the size of the database. So with our specific phenotypic method, we have these test microplates that are pre-filled with 95 different substrates that have been carefully selected to probe a particular metabolic pathway or a condition. And we also have a special redox sensitive dye in each well. And so when you inoculate this plate with a suspension that has the cells of the organism you want to identify, the metabolic activity of only the living active cells contributes to the dye reduction. And therefore, we're able to quantify the activity in each well. And so the result of that dye reduction is this distinct pattern that because we've selected a specific combination of substrates and compounds to put in the plate, should be unique for each species in our database. And we've divided our database into four distinct categories, and those are shown here with their respective plates. And we'll go through each of these in more detail a bit later. But you can see that aerobic bacteria, which encompasses both gram-negative and gram-positive species, anaerobic bacteria, yeasts, and filamentous fungi each have their own special plate. And in each plate, the combination of different compounds was selected specifically to be able to differentiate between species of that type of organism. So in each well, if the organism is able to use the substrate that's in there and grow or be metabolically active, then the dye will change color and get darker. And in wells where there are inhibitors, like perhaps salt or an antibiotic, if the organism can survive in the presence of that inhibitor, you will also see the well change color. And so to start using the plate, you just take your unknown microbe that's been cultured or streaked out on an agar plate, as shown in this left picture. You pick a colony and you dilute it into our minimal inoculating fluid, and then you just simply pipette that mixture into every well of the plate. And that process literally takes about 1 minute. And then you just incubate it and you read it, ideally kinetically. And the absorbance of the plate is then read, and the ID algorithm matches the pattern that's developing in the plate with every pattern in our database until it can make a good match. So this protocol is not just simple, but it's also compliant with GMP, and it can be used as part of a regular environmental monitoring process. And so to tie back to what I said at the beginning, what makes a good in-house environmental monitoring system? So we think there's three major factors. Ease of use in the middle here, which is necessary for being able to train quickly and get reproducible results. Second is the flexibility of throughput. So depending on your lab or your facility, you might need to run a few samples a month or up to hundreds or more a month. And then finally, the database is really critical to success because the more relevant species your organism can be compared to, the better chance you'll have of matching it. And so just to talk a little bit more about the Biolog database, because our products are intended for research use only, as well as veterinary diagnostic use, what you'll see that's not shown on this slide are any clinical applications for humans. So we do have many organisms in the database that are of clinical importance, but we've specifically designed our products for industrial microbiology. And this is different from some other products that were developed more for diagnostic testing, for example, looking at blood pathogens or other clinical applications. So as a company, our focus has always been on identifying environmental and industrially important organisms. So just to give you a few examples, we have Biolog present in many public health labs and large clinical labs, and the focus there is not on patient diagnoses, but rather on getting the correct ID on unusual organisms that can't be identified with a typical clinical system. Also for food and water here, we have multiple government municipalities around the world using our systems to verify that the water treatment is successful and that there aren't pathogenic microbes in the water. Over here for pharma and contract manufacturing, microbes, you probably know, are a significant threat to sterility of the product. And so it's crucial to be able to accurately identify microbes as part of your good manufacturing practice requirements that the industry sets. And we also actually have a lot of teaching labs over here for undergraduates that use these systems as a really great teaching tool for microbiology. So learning how to profile and identify different types of microbes. So you can see a lot of different applications here. And really, it's not just the sheer number of organisms in the database. It's really more about the relevance of those organisms to the application you're using it for. But we do have quite a large database. As you can see, there's over 1,500 aerobic bacteria, almost 400 different anaerobes, over 300 yeasts, and then notably over 700 molds or filamentous fungi. And we are continuously updating our database to add new species and update the nomenclature according to the latest widely accepted species names. And you can actually browse or search the current database on our website at biolog.com. So you can see a screenshot of it here, and we track, in each case, we track the version number. So you can, look at the most current version. And if you notice a species that's missing or you'd like to be able to identify, just reach out to us because we really welcome feedback from our users to ensure that we're adding the most relevant species that you want to identify. Okay, so before I turn it over to Max, I just want to spend a couple of minutes talking about our software and our instruments that are an important part of the workflow. So we have 3 systems for different throughput needs that allow you to choose the solution that's right for you. So the ID station on the left is kind of like a classic plate reader where plates are incubated somewhere else and then they're brought in to read one at a time. And so this system, like all 3 of them, are paired with our Odin for identification software. And so the software will automatically interpret the data and make the match. But you're reading the plates manually one at a time. The 2 Odin systems here on the right, Odin 8 and Odin 50, or Odin L, are fully automated incubators and readers, which means you don't need to move plates in and out of them. They will incubate the plates at the correct temperature, and then they'll automatically read plates, typically every 20 minutes, until they can call an ID. And they do that as soon as, it can call an ID as soon as it's able to. And the 2 Odin systems can also do a larger set of phenotypic profiling. So there's ways you can go deeper to kind of profile whatever organism you're interested in. And so they're able to do that work as well. So how do you decide which system is right for you? So there's some points here on this slide. In general, if we start on the left, if you have a smaller number of samples, or maybe you have plenty of incubator space in your lab, or you have incubators set to a bunch of different temperatures for different organisms that you're identifying, the ID station could be a great fit. Also, if you have staff, technicians on hand that are available to read plates at the specific time points, then that could be a great solution. If you foresee needing to run more plates at a time, or you need additional incubator capacity, maybe you need more incubators anyway, or maybe you're always short-staffed, like a lot of places are these days, and you'd prefer to automate the reads, then Odin systems are a really great fit. Because they're doing kinetic reads, you can often get the answer faster, and you also get all this additional phenotypic information about your samples. So Odin 8 is nice if you have typically one main user that's working with a maybe predictable or a regular number of samples. And then Odin L is really great for putting in a core lab or a facility that can have multiple users all coming in to use the instrument. And all 3 systems use the same software, which is called Odin software. And while the software and a lot of the aspects like the reports all look identical, the interface that you actually see when you plug in a particular instrument will change here. So here we're looking at a 50 plate layout for the Odin L or Odin 50, the 8 plate layout for Odin 8, and then this queue kind of system for the ID station where you make your list of plates and then you can just run them one at a time. So the software will just change the user interface based on which instrument it's connected to. And then, as I mentioned, Odin L is really accommodating for multiple users at the same time. So all the batches and the plates are organized within projects. And so as long as the temperature is set to what you want, any user can just walk up to the instrument, log in, and then reserve and load plates into any available slots. So you can see the free slots here. And so I could just pick those and put some plates and just start incubating and reading. And then the software also makes it very easy to quality check and export all the data. So because these instruments are all measuring absorbance at 2 different wavelengths, you have access to all of those absorbance measurements, and you can export them in a CSV format or JSON for further analysis that you want to do. And with Odin, as I mentioned, you get here the full kinetic curves for every well with information on what's in the well. So you get a, deeper view into the phenotype. But ultimately, what most people care about is just the report, right? A lot of times you don't care about the raw data. So you just want the report that has the ID. And Odin generates this very clear and informative report that can be saved as a PDF or printed. And in every report, there's always a results table here that tells you if it was able to reach a species level ID and also how long it took to make that call. So in this case, it took 4 hours and 56 minutes for this aerobic bacteria plate. And the table will also always show you the top 4 matched species. And it reports this value called a similarity index that's really an output of our proprietary algorithm. And you can think about that as the product of the probability, which is kind of like the separation between the top 2 species, and then the proximity, which is like the inverse of the distance between the species and the sample that you were interested in. So if both of these values are sufficiently large, then the similarity index meets that cutoff threshold. It's usually 0.5 for most cases, and then the ID will be sufficiently called. So here in this table, you can see the really large difference between the top match and the second best species. So there's a lot of confidence that the right species is the one that it called. But you can see that everything in the top 4 list comes from the same genus, which also, just gives a lot of confidence in the results. So the software also comes standard with many different security features that allow you to control the user access to the system and protect your data so that you can comply with your local regulations. And we also have an optional 21 CFR Part 11 package that provides some additional features like an audit trail, which you can see here, so you can pull up, the history of everything that's been done. And then it also enables electronic signatures of all the reports that you generate. And then one more feature I wanted to mention is that if you have species or even proprietary strains that you've made, for example, that are not in our distributed database for any reason, then we can actually help you add them to a custom database. So what I'm showing here is some screenshots from part of the workflow where you can enter in information about your organism. So you can enter in as much information as you want. If if it was something that came from a culture collection, or if it was something you engineered, or if it's just something that you found so you can basically enter in as much or as little as you know about it. And then we can help you generate this custom ID file. And then in the future, when you're running samples, you can actually decide whether you want to use exclusively your new custom database, or you search both the Biolog database as well as your custom database. So it's a really nice way that you can add things that are not in our database for whatever reason. And now I will turn it over to Max to go through more details on our different plates and protocols, and he has some great tips for working with each of those.
Thanks, Dina. So first off, I wanted to just mention these top 5 issues that we see a lot with microbial ID. I'm not going to go into too much depth right this second, but I will be talking more about each of these as I go through my part of the talk. So one of them is not following the instructions for use for the various plates that you're using. We have worked really hard to make these protocols as generally applicable as possible. So it's important that they're being followed to get the best and most reliable results. Also, using incorrect cell density to inoculate the plate, or using different growth media as your pre-culture instead of the ones that we recommend. And additionally, using uncalibrated equipment or starting with a culture that's too old can also really negatively impact results. Next slide. So the first plate type that I wanted to talk about is the Gen 3 plate. So this is for our aerobic bacterial identification for over 1,500 different taxa. And so this plate was created by screening through about 2,000 different phenotypic tests in order to determine which ones were the most useful in parsing microbial identity. And additionally, you don't have to use any sort of gram staining beforehand or any other diagnostic tests. As long as you know it's an aerobic bacteria, you can just go straight to the Gen 3 plate to start your ID attempts. Additionally, like Dina mentioned before, the workflow for this plate is very short. It only takes about a minute to set up. And it requires very few cells. So the way that the inoculation works is that you pick up maybe 1 or 2 colonies and then transfer those to a cell suspension to an OD of around 0.01, and then inoculate 100 microliters of that suspension per well of the plate, and that's it. That's the whole workflow. And so if you take a look at the graphic on the left here, we can see a few different sections on the Gen 3 microplate. So the left part of the plate is going to be nutritional utilization tests. So these are going to have different carbon sources, nitrogen sources, et cetera. And the amount of color development in those wells will indicate how well the bug was able to respire or utilize those substrates. And on the right 3 columns of the plate, these are our chemical sensitivity assays. And so we test for pH tolerance. There's a few antibiotics in there, as well as some tetrazolium dyes to look at reducing power. And between all of these 95 tests or 94 tests, we're able to determine identity. And so for some real world examples over here on the right, we have this Moraxella species versus Acinetobacter. And you can see the cyan circle, the well there, that's the negative control, and the pink one is the positive control. And so those are both correct on these plates, but you can tell that even though the Moraxella does not light up very many wells, we're still able to confidently call an identity, as opposed to the Acinetobacter, where nearly half of the wells in the plate are purple. So it really, it's not critical to have many wells be positive in order to have a confident ID. Next slide. So a few considerations to make when running a Gen 3 plate experiment. So as I mentioned, one of the pitfalls for getting IDs is to be using the correct pre-culture media. So in this case, for aerobic bacteria, most of the time you're going to want to use our bug blood agar. The BUG stands for Biolog Universal Growth Agar. And so this is validated to be as welcoming to as many different kinds of aerobic bacteria as possible. And it's very important to use the media that we suggest in these cases because it gets your bug into that correct and anticipated metabolic state before it gets transferred onto the Gen 3 plate. And the reason this is important is because sometimes we can call an identity as soon as just a few hours post inoculation. And so it's really critical to hit the ground running, as it were, in the right metabolic state that the algorithm anticipates. So that's why we thoroughly recommend that you use the pre-culture media that we recommend. For Gen 3, there's also 3 different protocols that are coupled with 3 different inoculating fluids. So the vast majority of species in the database are going to use protocol A with IFA. This is a pretty permissive inoculating fluid. The dye in it is fairly easy to reduce. And so most species are going to be identified with protocol A. However, if you use protocol A and you don't get an ID, the software will prompt you typically to then try protocols B or C, depending on what the results look like. So for example, if you have a plate that completely turns purple, including the positive control, that probably means that your bug grew a little faster than it should have on the plate because the inoculating fluid A was a little bit too permissive. And so in that case, the algorithm or the software will prompt you to use protocol B, which is more stringent and can limit the color development a bit more. And then if you have something that's very fastidious, doesn't grow very well at all, maybe didn't turn anything purple using protocol A, then we would recommend that you switch to protocol C. This is going to be a much more rich media. This is going to be more friendly to things that take a long time to grow or maybe have very small pinpoint colonies. A lot of gram positives are in this classification, but again, doing a gram stain is not required to get to this point in the workflow. The software will prompt you, or the instructions for use also have guidance on when to move from one protocol to the next. So additionally, when you're incubating the pre-culture plates, we require that they're incubated typically at 33 degrees Celsius prior to inoculation onto the Gen 3 plate. Again, this is to make sure that they're in the anticipated metabolic state. And then the cell harvesting is done very simply using one of our inoculators swabs. These have a 3 millimeter diameter, and they're made from tightly woven cotton. So they all you have to do is just touch the tip of the swab onto the plate on an area of dense growth, and that's typically just about enough cells to get you to the correct inoculating density. So we try to make it as easy as possible for you. And then again, when you inoculate the Gen 3 plate, it's equally critical to keep incubating at that 33 degrees Celsius, regardless of if your organism typically likes 36 better. We really recommend doing 33 because this is the generally pervasive temperature that most of our Gen 3 organisms need. And so if you're working with a true unknown, it's always best to start here. And then if you are using one of our endpoint read systems like the ID station, instead of using Odin, you can take reads at various time points. Here we have suggested doing 8, 16, and 22 hours, but just doing the 22-hour time point is often acceptable, and you don't need to worry about getting those earlier time points. Next slide. So the next major plate type that we have is our anaerobic or AN microplate. And so this is going to be for all anaerobic bacteria, which currently our database contains about 390 different taxa. And so this plate is set up very similarly to the Gen 3 plate, with the exception that it does not have resistance tests. It only has metabolic utilization tests, and with 95 different nutrient sources. And then this, for this one, the plate contains the same redox dye that absorbs at OD 590. So you'll see that same purple color development, and also requires a very low cell density, around an OD of 0.01. So a few considerations for this plate type. So this only has one inoculating fluid with it and one protocol. It's our ANIF, or anaerobe inoculating fluid. And to go with that, we also have our BUA auger. So this is an anaerobically packaged blood auger, and the inoculating fluid is also anaerobically packaged for your convenience. And again, just like with the Gen 3 plate, it's thoroughly recommended that you stick with the BUA unless you validate a different type of media. So with this protocol, you can use, again, no gram staining. It's inclusive of gram-negative and gram-positive anaerobic bacteria. And then these tests are going to be incubated at 36 degrees instead of 33 degrees. And similarly to the Gen 3 plate, the cells are harvested very simply and just transferred over to the inoculating fluid before being transferred to the plate. The one major thing that we emphasize with the anaerobic plate is that this should not be incubated in a traditional anaerobe chamber. And the reason for this is because many anaerobes, especially obligate anaerobes, will have very strong hydrogenase activity. And that can cause color development in the plate that has nothing to do with the carbon source in the well. And so we recommend that when you're incubating the AN microplate, you do so in a jar or anaerobe box system that just absorbs oxygen without producing hydrogen, and then reading as an endpoint at 24 hours, either in Odin or in the ID station. All right, so now that we've talked about the bacteria for a little bit, now we can get into my personal favorite, the fungi. And so the first one is going to be our yeast identification plate, or YT microplate. And so this is able to identify roughly 360 taxa at the moment, and is divided up into two different sections of tests. So the top 3 rows of the plate, these are going to be our, these wells contain the redox dye that we use and will indicate the respiration activity in those wells, whereas the bottom rows of the plate here in white, these are going to be reading biomass accumulation instead of metabolic activity. So you're still getting the same amount of phenotypic data out of here. We're just using 2 different types of tests. Next slide. Okay, so some more tips for these yeast identification plates. So we have a Biolog Universal Yeast Auger, the BUY here, that we recommend that you use for your pre-culture, and then the YT inoculating fluid that goes with the microplate. And when you are growing your yeast on the BUI, we typically recommend doing so at 26 degrees, although some cultures are physically incapable of growing at that low temperature. So 30 degrees is the upper end that we usually recommend for that. And then the cells are harvested just like with the AN and Gen 3 plates with that inoculator swab to the specified cell density that you can then transfer over to the microplate. And so with the YT and FF plates, we recognize that fungi grow a lot slower typically than bacteria. And so the workflow here looks a little bit different, where you can incubate kinetically in Odin, taking reads every 20 minutes, or if you're using ID Station for endpoint reads, you will take reads every 24 hours instead of every 8 hours. So you can just take reads at 24, 48, and 72 hours is typically the end for any yeast identification experiments. Next slide. So now we get into the filamentous fungi, or the fuzzy molds out there. And so this plate is our only microplate that uses a different redox dye than all the other ones. So this one is, instead of absorbing at 590 nanometers, is going to be 490 nanometers and is more of a pink color. It works functionally the same way, responding to energy production by the cells. But what it allows us to do is measure metabolic activity at 490 and then growth at 740 at the same time in the same plate. So we get essentially 190 total tests out of this one plate. And again, here we're not looking at resistances, we're just looking at metabolic substrate utilization. Next slide. So with the FF plate, there are a lot of idiosyncrasies that go with assaying filamentous fungi. And so to try to unify the protocol, we have one main recommended culture media, which is the 2% malt extract agar. However, we do also find success with potato dextrose agar as well. The key here is to induce spore formation or conidiation. And so that typically, again, we use the malt extract agar for and incubate at 26 degrees, anywhere between 5 to 10 days, depending on your organism. Until sufficient conidiation or sporulation is present to prepare the cell suspension, because we're going to be using spores instead of mycelia, if at all possible. There are some species out there that don't produce spores, and so those ones you can use hyphae or mycelia, and then just macerate those a bit and use that as your cell suspension instead in the FF inoculating fluid. And so these, similarly to the yeast plates, would be incubated at 26 degrees, either kinetically in Odin or endpoint every 24 hours in ID Station. And this usually takes between 1 and 4 days instead of 1 and 3 days like it did for the yeast plates. And the additional feature that we have for filamentous fungal identification is that sometimes it's very difficult for the ID algorithm to get to that 0.5 threshold for the similarity index that Dina was talking about previously. And it is necessary then to confirm the identity of your top match using morphology. And so we also have a companion macro and microscopic morphology library for our filamentous fungal ID workflow. So that way, if you're ever in doubt about your top match, you can go through our photo library and compare your culture appearance as well as some microscopic morphology, just to validate that your top ID hit is in fact correct. So this is an example of how that would work. So as Dina mentioned, the similarity index that we usually go for is 0.5. And so in this case, it's 0.491, but the rank 2 taxa is has a very low similarity index. And so we're pretty sure that the rank 1, this Penicillium chrysogenum, is the correct answer. But just to make absolutely positive that it is, we can then go to the photo library and check the morphology. And in fact, for this one, I ran this experiment. It was the Penicillium chrysogenum. The plate culture and microscopic images matched very well. And so we were confident in that identity. So as I mentioned before, we're expecting to use mostly sporulating fungi in the FF database. However, there are some that don't sporulate. And so for those that do, the typical workflow would be to moisten the swab with the FF inoculating fluid and roll that across the top of the culture just to pick up some spores and avoid getting any mycelial material, and then transferring that to the FF inoculating fluid. And it for the non-sporulating strains, those you can scrape some mycelia from the agar surface, being careful to try not to get agar because that can introduce additional carbon into the assay that we don't want. And then that can be mixed and macerated a bit. And then to avoid any excess bubbles or anything, you can then discard that supernatant and then resuspend in fresh inoculating fluid. Because the FF inoculating fluid has a gelling agent in it to avoid the cells settling down to the bottom while they germinate. Once you inoculate into the plate, the inoculating fluid gels and holds those spores or mycelia in suspension so that you have a nice even mix for measuring OD. Go ahead.
Yeah, so at this point, we were going to go back to our top five, and I've already seen a couple of questions come in that are hopefully have just been answered. There was one about preparing filamentous fungi for analysis. Um, but we were just going to review the top five and hopefully just touch a little bit more on some of these that we didn't do it yet. So I think Max went through, the sort of high level protocols for each plate. And I think one of the biggest things you see, Max, is that people will just skip steps or they, use a different agar. They think it'll be okay. But in fact, like you said, those protocols were really, very carefully designed. And so following them is really important.
Yeah, so the name of the game is the unified metabolic state here. So when we designed the assay and enter new species into the database, where everything is as unified as possible, using that same agar media, the same culture density, et cetera, in order to preserve that metabolic state, because we're quantifying the phenotypic output of these strains instead of just sequencing DNA, for instance. So it's really important not only that the bug is growing, period, but how it's growing.
Right. Yeah, that's a really good point. You talked a little bit about the OD, the cell density that's needed, in the inoculation at that first step. And so, you mentioned that the swabs make it easy because they sort of help you make sure that you have the right number of cells. But then maybe we also want to touch on the turbidimeter that we use and we mention in all of the protocols.
Yeah, so Biolog, all of our protocols by default use turbidity as the measure for cell density instead of OD or CFU per mil. And to make things nice and easy, the tubes of inoculating fluid are sized to fit in the turbidimeter. So all you have to do is take your tube of inoculating fluid, put it in the turbidimeter and blank to 0, and then do a quick check with one of the turbidity standards. There's one for each of the ID protocols at those specific turbidities to make sure that your dynamic range is measuring properly, and then put your tube back in and add cells to the desired inoculating density, and that's it. So using turbidity is a very easy way to be able to make your suspension to the correct density without having to do multiple dilutions, like you might if you're going back and forth between a 1 centimeter cuvette and a spectrophotometer over and over again.
Mhm. Yeah, that's right. And these turbidimeter systems come with every one of our systems. But if somebody does not have access to a turbidimeter for whatever reason, or they prefer making a different kind of measurement, we have some options here. Can you go through these?
Yeah, so we recognize that either there are people out there that don't want the turbidimeter, or maybe theirs is not operable at the moment, or something happens that you want to use some other metric other than transmittance for your cell density measurements. And so we have a couple of helpful technical notes out to convert between transmittance and either OD 590 or between transmittance and CFU per mil. And so you can use those values instead to generate your cultures.
Great. Yeah. So we've really tried to make it as easy as possible to start with the right density of cells. If I go back here, we We talked about the different agars, and I know, sometimes people who are working in different geographies have a difficult time purchasing some of the agar plates with the blood in them. Do you have any recommendations for what they can do there?
Yeah, so we have a couple of different options. So the first option would be that we not only provide the ready to go bug plus blood plates, but we also provide the base bug media as a dried media, as well as, and then you can just add defibrinated blood from a provider in your area to that powdered media and pour your own plates. And then additionally, you see here it says without validating first. So you can use other agar. We just really recommend that you go through several control strains first with your alternate agar to make sure that the identity results are consistent with the bug blood agar. And so a popular question that I get is, can I just use regular, my off-the-shelf blood agar that I've been using for culture? And sometimes that works. Sometimes it doesn't. It kind of depends on the supplier for the blood agar. We can't give a guarantee, and that's why we really recommend doing the validation first.
Yeah, that makes sense. So you could actually do an intentional validation for a new type of agar if you want. You can order some collection ID or type strains from a collection and you can run them and make sure that it works, right?
Yeah.
Great. Okay, so the next one on here hopefully is pretty obvious, but just using uncalibrated equipment. And I'm assuming that can mean a pipette, even like a multi-channel pipette or the turbidimeter or anything that you're using that's going to introduce errors.
Yeah, so especially when it comes to how much volume you're pipetting into each well. So depending on what plate type you have, for instance, the YT plates, those 3 rows that have dye in them, the dye is in the plate, not in the inoculating fluid. And so the volume in which you resuspend that the material dried into the well is really important to be able to get the correct anticipated color development. Because if you do low volume, then it will the color will be artificially high, and so it's important to just to make sure that your pipettes and other equipment are up to speed on their calibration.
Yeah, hopefully that makes sense to everybody who works in the lab. There's the number 5 here is starting with a mixed culture or an old culture. Can you talk a little bit more about the mixed part of it first? How do
Yes.
I know if I have a mixed culture or not?
Yeah, so the easiest way to determine if you have a mixed culture is if you have different morphologies. So the biolog plates are not able to handle mixed cultures and get a successful identity. And that's because when you have mixed cultures, you're getting the composite metabolic output of both of those cultures in the Gen 3 plate, for example. And the algorithm is just not able to parse that complex of data. And so it's really important to make sure that your strains are purified. And so the way that we usually recommend to do that is to subculture twice. If you're going from a mixed culture and trying to purify, to pick a single colony and subculture at least twice to make sure that it's been that your morphology is consistent. And you can also throw it on a microscope real quick to also make sure that the microscopic morphology is consistent as well. And additionally, the age of the culture is very important. So as you are growing your microbes on a plate, this is especially critical for some yeast and filamentous fungi, depending on how long they take to grow. As they accumulate cells on the plate, many of those cells in that colony are going to be either dead or static. And so when you transfer those to the inoculating fluid to generate your suspension, you have an artificially high cell density that's made up of mostly dead cells. And so we recommend that your cultures for use for bacterial applications are younger than 48 hours old. And then for typically we shoot for 24 hours for bacteria where possible. And then for yeast, try to keep it under 72 hours old. And then for filamentous fungi, as soon as they start sporulating and there's enough spores to use, run your identification experiment. Try not to wait for too long because some of those spores may start to become non-viable.
Hmm, that makes sense. Okay. Um, so we had talked about, and I see a question in the chat about using the FF plate actually for yeasts. And so I'm trying to interpret the question and say, is this a question of like, do you not know if it's a yeast or a mold? Or if you know it's a yeast, can you still go ahead and use the FF plate? And so I think we had a slide actually prepared for this, the first part of it, which is how do I know if I have a yeast or a mold? So maybe we talk about that first.
Yeah. So yeast and mold, the definition that we use for those is for yeast, you're looking for these small round budding cells producing smooth or potentially mucoid colonies on the BUI plate, whereas the molds, those are going to be those interwoven mats of hyphae coupled with growth at elongation tips instead of budding. And then most of these will form a fuzzy colony by themselves. But if not, looking for that invasive hyphae can also be an indicator. But really, the best way to make sure that it's a yeast versus filamentous fungus is to just do a quick light microscope look to make sure that it's budding cells versus apical extension for in the case of molds. And now for the other part of the question, can you run yeast on an FF plate? Short answer is yes, you can. But in general, most of the entrants from the that are yeast are only available in the yeast database, so your ID results probably won't be there. Now, that is with the exception of several dimorphic fungi. So there are those out there, such as some Candida species, for instance, or Cryptococcus as well, that have a yeast phase on some culture media and have a fuzzy or hyphal phase on another type of culture media.
Mm.
So for many of those, we do have those included in both the FF and YT databases. And so if you think you might have one of those dimorphic fungi, it would be good to just check the website for the database or to check on your software, you can access the database locally and see if your hypothesized sample identity is in both databases or if it's just in one.
Okay, yeah. But growing, I mean, as long as it grows on the right agar, like if a yeast is able to grow on that 2% malt extract, then you could get a phenotypic characterization of it. Is that right?
Yes, absolutely.
Yeah.
Mm-hmm.
Yeah, so I guess the answer is it is possible, and then just to get the ID, you'd want to just check and make sure that you're in the right database. Um, just sticking with fungi for a minute, because I know these are so interesting and they're actually pretty hard to identify with other methods. What are the challenges with identifying some of these fuzzy molds with other methods like sequencing or MALDI that
Yeah,
make
so the first thing is going to be the ability to use high throughput and easy DNA extraction tools like column kits.
Mhm.
Oftentimes, these filamentous fungi with their thick cell walls and heterogeneous suspensions that they make require some more hands-on methods to do DNA purification. And on top of that, methods like MALDI can often struggle because of lack of variation in surface proteins. And so doing phenotypic ID really has some advantages here of not having to worry about can I extract the DNA um, or purify anything else from the culture. All you have to do is make sure that your organism is growing.
Right, so you don't have to worry about breaking open the cells, extracting anything. It's just looking at the living, growing cells.
Yeah, and the other big issue that is present in sequence identification is just the lack of available reference sequences, especially at the level of strain identity. And so for phenotypic ID, we don't have to worry about having the correct reference genome to compare to. We just have the reference strain. And then as soon as that profile is captured and added to the database, it can then be used just like that for future identification efforts.
Yeah, that makes sense. I see a question here about, is there an oxidation control on the the anaerobic plate, or can we know if the plate had contact with oxygen or not? So I was just going to try to go back to that AN.
Yeah, so that's a good question. We do not have an indicator in the plate, but the way that the plates are packaged, they are packed with an oxygen absorber, and so when you receive the plates, they should look vacuum packed, as opposed to our Gen 3 plates, which have air inside of the package. So that's one way to make sure that the AN plates have not been oxidized. And then for the inoculating fluid, those do have an oxygen indicator in there. So as long as the fluid is not green, it means it has not been oxidized.
Okay, so yeah, I can look at the color of the wells and make sure that I haven't inadvertently exposed my plate to oxygen. Great. Okay, let me just look and see if you have, just a reminder, if you have any other questions, put them in now so we can make sure we have a chance to get to them. Okay, I see a question here. What might you suggest for pigmented bacteria where the pigments might absorb light near or at the dye area of the spectrum?
Yeah, so that's a good question. It is definitely a challenge to do to do identity this way for things that produce secondary metabolites like pigmented. Um, and so typically what we find is that bugs will ID pretty quickly in the assay. So if you're doing a kinetic ID, that would be my biggest recommendation for these types of bugs that produce pigment is to use kinetic ID. Because endpoint, you're giving them 24 hours in order to produce that pigment and potentially conflate the results. Whereas with kinetic ID, it gives a chance for the dye reduction to occur before there's significant enough pigment production. But generally speaking, the ID algorithm, it doesn't necessarily have to be dye reduction that the OD measurement indicates, right? So the algorithm is just looking for an OD 590 value. And so even if that there is some pigment production that is coincident with the dye reduction, that will be just recognized as an increased OD 590 signal in that well that is specific to that bug. So the ID should still work just fine.
Yeah, okay, so work fast.
Yeah.
And then there's a question here. This is a real tip and trick, I think. So if somebody has a really sticky bacteria, and the example they gave is a Campylobacter, do you have any tips for how to get a pure culture from something sticky like that?
Yeah, so getting a pure culture can definitely be challenging in those cases. Um, we also have, so in my experience with those types of things, like with Streptomyces, for instance, it invades the agar and the colonies are very hard and
Mm.
it's difficult to get it off of the plate. I found success with using the sharpened streakers that we sell and being able to really like dig into the colony a bit to transfer to another plate. But once you get it into the inoculating fluid, while bubbles are the enemy for when it gets into the inoculating or when it gets into the microplate, because those can cause issue with OD measurement. You can vortex and or otherwise agitate the culture once it's in the inoculating fluid. Just let it settle for a few minutes afterwards before you inoculate into the microplate.
Okay, that makes sense. Yeah, so you want to really make sure that you're picking a single colony, like you talked about avoiding mixed cultures, and then also yeah, just picking it with the right type of swab or tool. And we have a couple of different options. Okay, I'm just looking. There was yeah, so what happens if you can't find a species level ID, but you can get a genus level ID, for example?
Yeah, so the software will report a genus level ID,
Mhm.
or the other behavior that you would notice is that you'll have, like the Corynebacterium example that you showed earlier in the talk, Dina, the table of results, your top four matches will all have the same genus and might have and different species. And if none of the similarity indexes reach zero point 0.5, but they're all close to zero point 0.5 and all the genera are the same, then that would be sufficient evidence to call that as a genus ID.
Hmm. Yeah, which can still be really valuable for discriminating between different things. Okay. Oh, one last question that came in that might be worth. So it says, is it possible to read a Gen 3 plate, so again, the aerobic bacteria, after 24 hours if the negative control is still negative?
Yeah, so that's a good question. For the purposes of identification, there are very few strains, probably less than 50 out of the 1,500 in our identification database, that require incubation after 24 hours. I believe the maximum out of all of those is 30 hours.
Mm.
But the caution there is just, um, overdeveloping color can negatively impact your ID results, right? So that's actually why we recommend for ID station, if possible, you take reads, at 8, 16, and 24 hours. While doing it just at the end point is usually fine. Sometimes using those earlier time points can allow you to get a higher similarity index uh, sooner. And so incubating past 24 hours is not generally recommended unless the software prompts you with continue incubation.
Oh, right. Yeah. And actually, I mean, you have some clues just from your plate that you've incubated, your agar plate, right? Because if you can see that it's a really fast growing species, then you would sort of know that you'd want to check at an earlier time point. But if I
Yeah,
see small colonies, I might be okay waiting. If I get in late the next morning, I might be okay waiting a little bit over that, right?
yeah. You shouldn't your assay shouldn't fail if you are a couple hours late
Right.
on your last read. Yeah.
Yeah, so you've got sort of all these clues along the way from the agar plate and then how the plate's looking. But if you're reading it in Odin, the nice thing is you don't have to worry about the timing. It's just going to keep making reads kinetically. So great. Well, I think we're just about out of time, but I will just remind everybody that you will get a copy of the recording if you registered. And if you have other questions that you didn't get a chance to ask or that we didn't get a chance to answer, feel free to follow up with us afterwards. I have the email address here. And this was really fun to answer your questions and see which ones were coming in. So I really enjoyed it. And hopefully you got some interesting and useful tips and tricks for doing your microbial IDs. And thank you so much for your time and for joining us today.
Yes, thank you very much.